Hello everyone, welcome back. Last week we talked about staphylococci, and in the lab we would have used two different methods of growing them and differentiating. We have our Baird-Parker agar as the first one, which is going to be highly selective due to its potassium tellarite and lithium chloride. These two components are going to allow the Staphylococcus aureus colonies to appear grayed to jet black with a white margin, and they'll each be surrounded by a clear zone, no pig zone, okay? Then we have our staph epidermidis, which will be gray to jet black with no clear zone, and there will be very weak growth in comparison. This is a picture of staph aureus right here. Then we have our biochrome staph aureus agar, which is going to be a commercial product. So if you're going to be testing a large amount of food products, you could buy these pre-made in a large set, and a positive result for staph aureus would appear these mauve colonies right here, whereas epidermidis will have no color and very weak growth in comparison. Lab 8 is going to be talking about two different genera, Bacilli and Clostridia. The first one, Bacillus, gets its name because all of the organisms are rod-shaped bacillus organisms, and they're also going to be gram-positive and spore-forming, but they will be either aerobic or facultative anaerobes. They're going to be a little bit tricky. And then they will be found in soil, and they're going to be causing a lot of contamination for food and plant products, as well as foodborne disease for some species. The first one we're going to be talking about is Bacillus subtilis. This is aerobic, so it can use oxygen. And then we are going to be finding it in soil, and this organism, subtilis, is perfect for studying sporulation in the lab, and it's not going to cause any illness. It's not non-pathogen. Whereas Bacillus cereus is going to be facultative anaerobe, so it uses oxygen, but it doesn't have to use oxygen. And then also be found in soil, but this one is going to be causing major symptoms such as diarrhea, nausea, and vomiting. So this is a big concern for leaving any food out in after heating it up, cooking your rice, and then you leave it out for several hours. Then you can give it time to produce these toxins, and then you can get sick by eating it later on. For all of these organisms that produce spores, we can do something known as a spore stain, where the cells will be dyed pink and the actual spores are going to be dyed green, so you can differentiate between them. So if the organism you're using the stain on didn't produce spores, it would only be the pink cells. There would be no green spores. The next genus is Clostridium, which is also going to be gram-positive, rod-shaped, and spore-forming, but in this case, they're only going to be anaerobic. Very similar habitats as well. Clostridium perfringens is the first species. This is going to be anaerobic. Again, all of them are going to be. And found in soil, decaying vegetation, inside of our GI tract. And this is going to be a pretty serious organism because it produces an enterotoxin, which is not going to be fun. It's going to cause intense abdominal cramps, bacteremia, gas gangrene, diarrhea. It's not a good thing. Then we have Clostridium botulinum, which is an even bigger problem. First isolated in 1896 by Van Emmengem, found in soil as well. Most of these are today. This is mainly associated with food poisoning, especially for canned foods, because that creates a good anaerobic environment for these organisms. And Clostridium botulinum is going to produce the Clostridium, the botulinum neurotoxin known as botulism, which is a serious paralytic illness. And only 10 grams of that neurotoxin is a lethal dose for the entire population of Los Angeles. So any labs that are going to be working on this neurotoxin do require a special permit, and Cal Poly does have this, and Dr. Lynn's lab she doesn't work with the actual neurotoxin, but she does work with Clostridium botulinum. And Clostridium sporogenes is going to be our closely related non-pathogenic strain. So this is going to be the one that isn't going to be causing any illnesses. So with that, I hope I was able to answer any questions you guys have within your emails. If you have any more emails, please let me know. This week you're going to be posting your video presentation, so I'm looking forward to seeing that. I've looked through most of your PowerPoints and they look really good. I'm very impressed. So keep up the hard work. Keep on going. You're all doing great.